Americanize their scone.
Yeah, I don't know why when you get a scone out, it's so dry.
I hate dry scones.
It doesn't have to be that way, it just is often.
People in Bid, Missouri just got on Fox News.
What's that?
Apparently, like, the couple from Bid, Missouri was operating a illegal like animal rescue. Turns out they were like, they had like euthanized 300 animals. They were killing like 150 more.
Oh my god.
Yeah.
Lots of drag. Alright,
Only in
but
Bid, baby.
news is over. It's time for genetics. Alright, so I'm on the Moodle site. I had posted the National Academy of Sciences genetically modified organisms report on there. Hopefully you've been reading chapter 3, which everybody needs to read. Have you been reading chapter 3? Okay. If you haven't, start. New set of food blogging prompts are up. This is the third bout of food blogging. Just 2 topics this time around. One is genetically modified organisms. Today we're going to talk about genetically modified organisms and how prevalent they are in our food supply. Even if you don't think you're eating them, you're eating them. So one set of prompts has to do with that. The other set of prompts actually is a little funner. It has to do with food fads. Monday was the first day of fall, and it's pumpkin everything. If you go to a Quick Trip, there are pumpkin pretzels. I have no idea why you would want to bastardize pretzels like that, but it's like pumpkin spice everything. And this is something that used to not be a thing. This is a relatively recent sort of fascination people have with making the world pumpkin spicy at this time of year. So I ask you in a couple of the prompts to reflect on food fads. What are the kind of food fads that you've adopted as not just a fad, but it's now become part of your habitual eating habit? Or do you think food fads are just totally bogus and you avoid them like the plague, like I do? The food blogging prompts are there's only 2 of them. Comment on that topic twice over the 2 weeks. I've posted today's slides. We're going to finish up the last little tiny bit of kind of Mendelian genetics, and then we're going to be talking about genetic engineering, getting ready for you being able to handle the things that are in the National Academy of Science report. And then I've also made an assignment for you for which chapters you will be dealing with. So for chapter 4, it's Riley, Charlie, Sydney, and Noah Espinoza. Chapter 5 is Dakota, Noah, Caden, and Darren. And chapter 6 is Thomas, Minnie, and Brandon. So I just divided you up equally. So those are the people that you'll be working with to prepare a presentation. Be ready for that presentation on October 4th. We're going to finish genetic engineering long before then. We'll actually probably be finished with genetic engineering on Friday. But I wanted you to have some time after we talked about genetic engineering in class to prepare for making a presentation. It's a 15-minute presentation that basically is just summarizing what the information was in your chapter. We have 3 groups. That'll be about 45 minutes, and then we'll spend the rest of the time in that class talking about that report. And this is all being done in preparation for you writing a position paper after October 4th. I'll figure out between now and then when I want that due, but it'll probably be after fall break, but before November, more than likely, so that we don't leave that too late into the semester and then things start stacking up on you from other classes and things like that. So anyway, be prepared for that by October 4th. You only have 1 chapter that you need to prepare for. I'm covering a certain amount of the information that is in chapter 3 over the next 2 days of lecture, and I can fill in anything on that day on October 4th from chapter 3 that you might have questions about. So those assignments, they are up. And that is that. Any questions about that? Yeah.
So just to be clear, we only have to post twice in 2 commas in the next 2 weeks, or
So you have 2 prompts,
Yeah, so-
post on each of them
twice?
twice
Okay.
over the 2 weeks. That will be your 4 posts, and then you still have your commenting and things on other people's things. My guess is you're probably going to have quite a lot to say about people's food fads, and what they've adopted and not adopted. Like, you know, I consider a lot of the things that appear at Taco Bell periodically to be food fads because they cycle through their menu relatively often, adding new things, taking things away, etc. Yeah, Brandon.
What is a food fad exactly?
Oh, like pumpkin spice everything. Like right now, like all of a sudden it's fall, so everybody's putting pumpkin spice latte at Starbucks. I'm assuming they probably have that now, but that's a seasonal thing. It's not something you can't get a pumpkin spice latte like
In the summer.
all throughout the year. Yeah, you can only get them for this limited sort of time. And that's a thing that if you had gone to Starbucks 10 years ago and said, I would like a pumpkin spice latte, they would have been like, what? We don't have that. So this is kind of the definition of a food fad. Something that comes up, is super popular, and then hopefully someday goes away. Pumpkin spice hasn't yet gone away, but maybe it will at some point.
Why do you hate pumpkin spice so much?
Well, I mean, for one thing, if you look at the ingredients in pumpkin spice, there's no pumpkin in there anywhere. It's all
It tastes
stuff like
pumpkin, though?
it's all stuff like nutmeg. Because if you taste pumpkin, so
I know it'd be overpowering. and
So
a
here's
little
the thing.
nasty, but
go to the store this week, homework assignment, ungraded, but go to the store, pick up a can of canned pumpkin puree like you would use for making a pumpkin pie. And look at the ingredients. You know what you will find?
No pumpkin.
You will find no pumpkin. It's other kinds of squash in there. You know what makes it taste like pumpkin?
Artificial flavor.
Not artificial flavoring, but spices. They put spices in there that give you that pumpkin flavor. But in many cases, the squash, so pumpkin is a type of squash, but in many cases, the type of squashes that are in canned pumpkin puree that you would use in your pumpkin pie is not actually pumpkin. It's like butternut squashes and other kinds of squashes and pumpkin spice. So this idea of what we taste as pumpkin isn't actually pumpkin at all. It's like nutmeg and cinnamon, things like that. That's what pumpkin tastes like to us.
okay, would you be okay with it if it was named anything else then? Is it just the name that is bugging you?
So I don't like the falsehood of the name, but then also I just don't like these bandwagony kinds of things.
It's like seasonal. I mean, it's seasonal. It's seasons. I mean,
So-
who's gonna drink his
so
pumpkin
anyway,
spice
that's-
in the summer?
So that's a fad. Let's see, what are some other fads? Sometimes diets are faddish. So when I was in graduate school, one of my professors was on the cabbage diet. He ate nothing but cabbage. I'm like, really, nothing but cabbage? And then he just kind of got pale and sickly looking, and he smelled bad all the time. And the fact of the matter is, he didn't lose any weight, as it turns out. That fad came and went.
Red
that-
meat diet?
Huh?
What about the red meat?
Yeah,
diet?
so like the Atkins diet, that was very fatty. That came and went. Poland talks in the book about how we demonized bread for a period of time because carbs were supposed to be so bad for you, et cetera, et cetera. So fads are food fetishes, if you want, that arise and stick around for a little while and then decline. And in some cases, they catch on and they go from being a fad to being a part of our food culture. And in other cases, they come and they go. And there's just a small population of people who continue the fetish because they took that fad on and made it part of their normal diet. Yeah.
So I just looked up pumpkin pie cans on
Yeah.
Walmart.com, and all of them have pumpkin in them.
Do they have other?
So this just has pumpkin.
Oh, because that's Libby's, it's 100. Okay, so why are they advertising that they're 100% pure pumpkin?
Because others,
Because others do not.
great value even is.
Is Great Value even?
Mm-hmm.
Oh, give me Great Value. Let me see. Is it?
It's out of stock, but.
Scroll down. See, but what's not? So that's a product description? Oh, well, that's the ingredient list. Okay, all right. All right. Well, maybe they've changed because 5 years ago you could go and get pumpkin at the store and it was other kinds of squashes. And maybe it's people like me railing against the pumpkin industry that has changed things.
Well, now you can,
Huh?
now you can participate in the.
Now I can
seasonal-
participate. Now I can make a pumpkin pie if only anybody in our house liked pumpkin pie. Actually, I like pumpkin pie, as it turns out. I also like pumpkin bread. But you know what we used to do when I was a kid? We would raise our own pumpkins, and then we would use those pumpkins to make pumpkin pies and pumpkin bread.
Pumpkin donuts.
Hmm?
Pumpkin donuts.
I've not had, see, that's another thing, pumpkin donuts. Pumpkin donuts were not a thing.
What
Yeah.
season did you make your pumpkin bread?
Well, during this season, because this was when pumpkins are maturing. It takes all year to grow a pumpkin.
Winter season.
So anyway, food fads, it doesn't have to be the pumpkin fad, it can be anything. There was a time when, you guys know who Paula Deen is?
Yes.
Yes.
She has a recipe for a hamburger, and it's basically a hamburger patty, and the buns are 2 Krispy Kreme donuts. That was super fatty for a while. Everybody was eating those things. It sounds like a heart attack waiting to happen.
You can find that stuff at like the fair too.
So there a lot of times food fads appear at things like state fairs, and you have a deep fried Snickers bar at the state fair. Do you then come home and adopt deep fried Snickers bars as part of your normal diet, or is that just like state fair food? And there's also this idea of contextual eating. I will do contextual eating a lot of times. So if I go to a baseball game, you've got to get a hot dog and a beer. I mean, it's not really a baseball game unless you're eating shitty food that's way too expensive at the ballpark. It's just a thing. Go to the ballpark, get a hot dog. I bike ride. There's a taco ride. It rides from taco stand to taco stand throughout Kansas City. That day, I'm eating tacos. You ride 40 miles and probably consume a dozen tacos over 40 miles. Feel miserable at the end of it, not because it was so long, because it really wasn't that long and the pace wasn't that big, but you've had a lot of tacos and probably a healthy amount of beer in the process of going on that ride. So there's also contextual eating, but I'm thinking more like fads that have come and gone. Which ones did you take on when they were fads and then dropped off when the fad disappeared, versus which ones have kind of stuck with you? So anyway, that's that. All right, last couple of slides on genetics. I know you guys are tired of genetics. So the reason that we're going for all of this is for a couple of reasons. One, when farmers plant their crops, they almost always plant hybrids. And they have to buy hybrid seed every year. So they're kind of, they're kind of stuck with big agriculture, Monsanto, Cargill, Archer Daniels Midland, these places that produce hybrid seed. And it's because hybrids have a combination of traits that generally increase yield. And the big ag companies know this, and they have enough money and enough land to grow hybrid seeds, because to grow hybrid seeds, it's fairly labor intensive. To get enough seeds to sell to farmers, you have to do this on a really large scale, and the only people who can do this are large corporations. And so this is a figure from your textbook, basically showing you how doubly crossed corn is constructed. What they do is they take corn plants in a field and they inbreed those corn plants over a number of years so that they're homozygous for all their traits. And they choose plants that have a particular genetic trait. It might be herbicide resistance, it might be drought resistance, it might be any trait that they're interested in. But each of these strains of corn is then homozygous for that trait, which means it's true breeding, which means that if you breed it to itself, it will produce the same phenotype that the parents have. And then they cross those plants. And when they cross those plants, it will be heterozygous for both of the traits that are desirable. It'll be heterozygous for the trait that is in the inbred plant B, and it will be heterozygous for the trait that is in inbred plant A. But then they won't cross them again with one another. They then cross them with another strain that also has other traits that are desirable. And what you get in the end here is you get a bunch of plants that are heterozygous for all four of these traits, presuming that all four of these traits are traits that are desirable for increasing yield. But because they're heterozygous for all four traits, it doesn't pay for a farmer to allow those guys to breed with one another, because you're going to get a lot of homozygosity out of that breeding because they're heterozygous for all of these traits. So if you have trait A, B, C, and D, these final plants down here are heterozygous for all of these traits, which means that they all express this particular trait. But if you cross these with one another, this is now a really complicated cross. It would be 128 cells in the Punnett square. Not something we're really prepared to do in class, but we have done this for just 3 traits. So let's go back and look at that. We did this for 3 traits, and when we did this for 3 traits, you don't have any of the desirable traits in this plant. You only have one of the desirable traits here, one of the desirable traits here, one of them here, one of them here, 2 of the 4 desirable traits here, 2 of the 4 here, 2 of the 4 here, only 1 of the 4 here, 2 of the 4 here, 2 of the 4 here, 3 of the 4, 2 of the 4. There's big chunks of the offspring that come out of this that don't have the desirable traits. And so a farmer buying hybrid seed from Monsanto or something, and then saying, oh, I'm gonna save money next year by not buying hybrid seed again because hybrid seed is expensive. I'm just gonna let these guys reproduce with one another. You would get a big chunk of your offspring that have less than all 4 desirable traits in it. And so rather than farmers saving seed, so back before hybrid seed was a thing, farmers would just plant their crops, they would sell some of the crop, and they would hold some of the crop back to seed next year's crop. And that was cost effective because you're not paying somebody else for seed. Once you plant a corn crop, you are in the corn business from then on. But when there came a stress to get higher and higher yields from your corn crop, certain traits promote higher yields, and the best way to get those traits in your plants is to do this process of hybridization, starting with recombinant inbred lines that are homozygous, crossing those, and then crossing those with other plants so that you have all of the desirable traits. But because all of those desirable traits are in the heterozygous condition, you get a big chunk of offspring from that cross that don't possess the desirable traits. And so the farmer, rather than allowing his crop to self-fertilize and saving back some of that seed that he plants next year, over half of that is not going to have all 4 desirable traits. And as a result, he's better off actually just buying hybrid seed from Monsanto year after year after year after year if he hopes to sustain the kind of yields he gets the first year that he plants the crop with the desirable traits. And so hybrids have increased yield, but they have come at a cost, and the cost is the cost of the seed, which is usually very expensive. And the reason that it's very expensive is because it's very labor intensive, time consuming, and takes a lot of land in order for these big agricultural companies to produce hybrid seed. There are some farmers who actually are in the business of producing hybrid seed. And so they don't produce seed to put out on the market. they're hired by Monsanto or Cargill or Archer Daniels Midland or whatever to basically set up a set of fields where they basically control who gets to reproduce with whom. And that's all they do. When they're done, they sell their crop to Cargill or whoever, and then that's the seed that goes out on the market the following year to be sold to farmers, this hybrid seed that has these desirable traits that increase yield. And it all comes back to, when you cross heterozygotes, you get a lot of dilution of the dominant traits in the offspring because of just simple Mendelian genetics acting against you. So, whoa, what happened? So this is the segue into the next presentation, which is a pug and a corn plant. I'm almost finished with finishing the key to the basic genetics problems. I'm working on the last one, getting it formatted. I'll probably have it out in the next 45 minutes to an hour after we leave class. It's up on my screen actually. It is right here. I just have, I'm working on the last question, getting the answer typed up. So I'll post that so you can practice a dihybrid cross, and that will help you hopefully on the next exam. Questions about basic Mendelian style genetics? Okay, because now we're going to a place that Mendel could never have conceived of us going. And why it's not opening. Okay, that's weird. Okay, so when we want to talk about genetic engineering, I want to clear up some terminology, because the National Academy of Science report deals with GMOs, and GMO stands for genetically modified organism. I would argue that you can be genetically modified in a variety of different ways. One of the ways you can be genetically modified is through selective breeding, artificial selection, which is what we had talked about before when we talked about selecting lines of corn for increased corn oil production. So artificial selection is one form of genetic modification, and it's the kind of modification that allowed us to go from wild wolves to a pug. We didn't genetically engineer a pug. What we did was we selected for the traits that would lead to ugly dogs with respiratory problems, and we got ugly dogs with respiratory problems. Sorry, do any of you own a pug? Okay, I feel so sorry for pugs, because we've selected for changes in cranial structure that actually makes it difficult for them to breathe. And this was all done simply by people selecting dogs that had particular features. There's this experiment that was done actually it's ongoing in Russia, where this Russian lab got a bunch of foxes, and basically they selected for one trait. The trait was whether or not the fox seemed willing to have interactions with humans that weren't biting, aggressive interactions. So they basically selected foxes for future breeding that were more docile. And so they basically selected for docility. And when they selected for foxes that were docile, all of these other phenotypic traits got pulled along. Their ears, rather than standing straight up, got floppy. The head structure changed. So the genes that evidently control personality in dogs are also linked to a bunch of morphological characteristics as well. So this was all done just by selecting certain dogs to breed and preventing other dogs to breed. This is how we got pugs, this is how we got Great Danes, this is how we got the Chihuahua, was through selective breeding. In the same way, we started out with teosinte. Teosinte is believed to be the precursor to modern corn, and through selective breeding of teosinte, we went from this thing, which is, you know, a single stalk with seeds that shatter, seeds that are released from the plant, to this thing that has tons of kernels. The kernels are all wrapped up in sheaths of leaves, such that the seeds cannot escape. Once you get the corn cob out with all of the kernels on it, those kernels are firmly attached to the cob so that they don't come off easily. This is all because we are preserving this thing for crop. We want those seeds to be intact because we want to eat them or plant them or whatever else. And so this went to that, but that wasn't through any sort of genetic engineering. That was just through selective breeding, looking at teosinte, looking at the variability of teosinte, and selecting varieties of teosinte that had characteristics that humans found useful. That's different than genetic engineering. So when the National Academy of Science puts out a report on GMOs, they are talking about genetic modifications that in biology we would refer to as genetic engineering, not simply selective breeding, okay? So genetic engineering involves taking a gene out of one organism and putting it in another organism using the techniques of molecular biology. And so when we talk about genetic engineering and genetically modifying organisms, the way the National Academy of Science is talking, that's what we're talking about. Occasionally I see memes on places like Facebook, where I have friends who don't know anything about genetic engineering, and they're like, a visual guide to identifying genetically modified corn, non-genetically modified and genetically modified. The thing that I kind of like about this meme is it's kind of one of the deeper memes out there about food, because yes, this is non-genetically modified corn and this is genetically modified corn. Now this corn might also be genetically engineered, but the differences between this and this are just differences in 7 genes. Alter 7 genes and you can change this plant into this plant, because these are genes of large effect that have large effects on the structure of the corn plant. But all of these 7 genes that get modified through selective breeding, those are all genes that were in the teosinte plant originally. And you just selected for different variations of those genes to produce modern corn. And so it's poking fun at people who are worried about genetic modification, but in the process of doing that, they're actually talking about genetic modification in the term of natural selection, which is not the same as genetic engineering, which quite frankly is what people are concerned about. And so, complicated explanation of a stupid meme. So, we have a lot of crops on the market that are approved for growth and use. First, genetically modified organisms that were approved for use, I think it was 1996, I've got it written down later on. But the modifications that we have for plants oftentimes have to do with either herbicide resistance or pest resistance. In some cases we have genetic modifications that are designed for drought resistance. So we have alfalfa and canola. We use canola to get things like canola oil. In the case of these two, you have herbicide resistance. In this one, it's actually a component of the oil that is basically upregulated through genetic modification. In corn, we have BT corn that, BT, whenever you see this term BT, this abbreviation BT, this stands for Bacillus thuringiensis. This is the scientific name of a bacterium. And it's a bacterium that kills insects. It's normally found in soil, and they've taken a gene out of this bacterium that occurs in soil, and they put it into plants. The plants then produce this toxin, and the toxin is active at killing insects. As it turns out, because the insect physiology is so much different from our own physiology, that the operation of the Bt toxin doesn't have any effect on us because it deals with, it messes up insect developmental biology, and our developmental biology is very different. So we have Bt for insect resistance and herbicide resistance. Whenever we're talking about herbicide resistance, almost always we are talking about resistance to Roundup. Roundup is a name brand of a herbicide that contains glyphosate as its active ingredient. You can go down to the hardware store, Lowe's or Home Depot, or any garden center and buy a thing of Roundup. You put it in a sprayer, and you can spray the weeds in your yard. You can spray the weeds that are coming up through the cracks in your driveway, and it kills plants, and it kills plants really effectively. And they found a gene that will impart resistance to Roundup. So if you're a farmer and you want to control weeds, when a farmer plants a field, he plants the plants in rows because it makes it easy to harvest them and fertilize them and everything else. But what you don't want, so in these rows you have your corn stalks coming up. What you don't want is weeds coming up in between the rows, because they're going to be stealing water and nutrients and things from the soil that could go towards growing your corn. Well, in the old days, when you wanted to get rid of these weeds, one of the reasons that you planted things in rows is you would actually go through and plow between the rows of corn or the rows of soybean or whatever. And as you plow, it would upend the weeds that were growing in the rows and kill them, because their roots would be exposed, they would dry out and die. But in the process of doing that, it causes a lot of soil erosion. Running a tractor over and over and over your field also leads to compaction of soil. The other problem with upending the soil like this is not only does it cause more erosion, but it also opens that soil up to drying, so you then have to irrigate it more. So there are lots of problems with using plowing as a method of weed control. Not only that, it only gets the weeds that grow up in between the rows. If there are weeds that are growing right next to the base of your corn plant, there's nothing you can do about that except to go out there and hand pull them. And who's going to go out to a 2,000 acre cornfield and hand pull weeds at the base of corn? If, on the other hand, you can spray an herbicide on your field and it will kill the weeds and leave your crop standing, you get rid of all of the weeds in the rows, and you get all of the weeds between the rows, and you get rid of all of the weeds that are within the rows that you couldn't have plowed up anyway. As a result, you get a weed-free crop that you don't have to plow. You don't have to water it more because of the plowing aerating the soil. You don't have to do a lot of things to it. You can just spray it with an herbicide. The weeds that don't have herbicide resistance die, and the crop that does have herbicide resistance is left standing. And so that's the reason that we have herbicide resistant crops is basically so we can spray herbicides on the crop won't die, and the weeds will. Also in cotton, you have the Bt gene. Papaya, there's virus resistance. So this is just basically a bunch of the crops that we have some form of genetic modification. In all of these cases, genetic engineering, in all these cases of genetically engineered crops, you've taken a gene from some other organism and you've inserted it into the genome of a corn plant or a tomato plant or whatever. In some cases, the genetic modification isn't actually that. In the case of tomatoes, the genetic engineering part is that they've gone into the tomato and they've basically removed a gene that causes the tomato to ripen, and they've slowed down the ripening process because tomatoes are interesting. I like tomatoes a lot. My tomato crop this year has not been great. But with tomatoes, the problem with tomatoes is when they're ripe, they're soft, which means that they don't pack well, which means that they don't ship well. And so what you generally do with tomatoes is you harvest them before they ripen, you pack them up in boxes and you ship them, and in their unripened state, they're firm, which means it's easy to pack them, easy to ship them, and they won't get damaged. The problem with that is by the time you harvest it, pack it, ship it, across the continent or to another country, if it ripens too fast, it gets to the destination, it's ripened, it's gotten soft, it's gotten jostled around, and they're bruised and damaged and everything else when they get to market. And so the first plant that was brought on the market was a delayed ripening variety of tomato so that you could pick it early, have it go through extended shipping and still arrive not yet ripe so that it wouldn't be damaged in shipping, and so it delayed ripening. And they didn't do that by inserting a gene, they did that by just turning off one of the genes that is naturally in tomatoes. Thicker skin, I actually do not like thicker skinned tomatoes, which is why I like homegrown tomatoes, because I just don't like that thick skin. But once again, the thicker skin just makes it easier to transport and pack tomatoes. So, if you look at the amount of acreage that is planted in genetically engineered versus non-genetically engineered organisms, you see that there's actually quite a lot of genetically engineered soybeans, a lesser degree in terms of percentage of total area, but still quite a lot of area. When you look at the different kinds of traits, a lot of these traits are actually herbicide resistance, followed by insect resistance or a combination of both of them. And so when you look at, and we'll see some graphs here in a minute of kind of how this has changed over time, it's quite surprising to the uneducated and unexposed how much of our food supply is actually populated by genetically engineered organisms. So in 1982, the first genetically engineered plant was produced. It was an antibiotic-resistant strain of tobacco. And this was basically just a test of concept. They wanted to see if they could do it, because there's no reason for tobacco to need antibiotics, really. And so the reason that tobacco was used was because there was a virus called the tobacco mosaic virus. And the way a tobacco mosaic virus operates is by injecting its genes into the leaf cells of a tobacco plant, and then those genes insert themselves into the genome of the tobacco plant, and then the tobacco plant just uses, in the process of replicating its DNA, it also replicates the viral DNA and creates more viruses, and this is how viruses spread. When you get a virus, when you get the flu virus, the influenza virus, it basically injects its DNA. That DNA gets incorporated into the cells into your genome, and then every time you go through an episode of cell division or replicating DNA for whatever reason, it basically makes a copy of itself, and these things kind of hijack the cell's genetic architecture. So 82, antibiotic-resistant tobacco. 86, a useful trait, herbicide-resistant tobacco in 1987. BT tobacco, once again, having this toxin, Bacillus thuringiensis. These two traits actually help increase yield of tobacco. Yeah?
So you said at the beginning that it takes a long process for them to make this one crop homozygous.
Yes.
How long does that actually take? Is that
So this isn't done. This isn't done that way. This isn't being done through selective breeding. This is basically finding a gene in one organism that does a particular thing, saying, oh, that's useful. We're going to now insert that gene into the genome of tobacco in this case. This is not being done through selective breeding. This is done through genetic engineering. So this is not hybridization or anything else. This is,
So there's no selective breeding in the, like, molecular process of
nope.
changing
You
genes?
clip a gene out of one organism and you insert it into the genome of another organism. And so in that way, it's much quicker and much more efficient in terms of modifying the phenotype of the crop that you're growing, because it's just molecular biology in a lab somewhere. Inserting genes into, and we'll see how this is done here in a few minutes. So let me get through the history, and then I'm actually gonna show you how it's done. There's a different type of bacterium that also infects plants that we do a lot of this with, and you'll see how you can open up DNA, get a gene into that DNA, and then use a bacteria to get that gene into a plant. So by 1984, we had the first GM crop approved for sale in the United States. It was this flavor saver tomato, this slow ripening tomato. So I was off by a couple of years. 1985, there was a Bacillus thuringiensis potato. It's an insecticide producing crop, followed very quickly by canola, corn, and cotton later on that same year. 1997, we had herbicide resistant wheat. 2002, Monsanto applied for a patent. They were going to be marketing this herbicide-resistant wheat to a lot of foreign countries, and a lot of foreign countries didn't want it, and so they pulled plans to market it because of objections from Japan and South Korea. So in some cases, countries have said, we don't want your genetically engineered seed, and that was enough for Monsanto to pull it off of the market. First perennial GMO crop was, genetically engineered crop was approved for use in the US. It's a herbicide-resistant alfalfa. One of the problems with a perennial is that it's now a genetically engineered perennial, and a perennial grows year after year after year. It's not an annual plant. Like corn is an annual plant, soybeans are an annual plant, tomatoes are an annual plant. You plant it in the spring, it grows through the summer, it produces its crop, and then it dies. So there's less of a possibility of those genetically engineered modifications getting out into other organisms because the plant doesn't live longer than a year. So it was big news when the first perennial crop was approved. By 2013, 87% of soybeans are genetically engineered, and you can see the rise of genetically engineered crops over time from kind of shortly after they were first approved. Soybeans is now over 90% of soybeans are genetically engineered for herbicide resistance. Cotton for BT. We have BT corn, which is at about 80% of the crop. So your sweet corn that you eat from the grocery store, chances are that sweet corn has not been genetically engineered, and that makes up the other 20%. This is all corn that is going into the commodity market, corn that is used for making cornmeal, corn that is used for making ethanol, corn that is used for making plastics, and all these other uses of corn. Most of the corn that you get that is sweet corn is not going to be genetically engineered. So this basically shows that the rise of this has happened, and for most crops, 80% or more of the crop that we have has been genetically engineered in one way or another. So how do we do this as biologists? Nowadays, we have a variety of different ways of doing this. One way is to use the genetic toolkits that other species have evolved. So Agrobacteria tumefaciens, this is a tumor-inducing bacterium. So it gets on a plant, and when it's on a plant, it induces tumors in those plants. So you have excessive plant growth, galls forming, and things like this. And so in bacteria are weird. They're different from us in that they have a circular chromosome. Remember that our chromosomes are linear. Bacterial chromosomes are circular. They're continuous. And in addition to the regular bacterial genome, there's also usually something called a plasmid, which is a small section of DNA. It's also circular, But plasmids are able to be transported out of the bacterium into another bacterium, or in the case of Agrobacterium tumefaciens, it takes that plasmid and will actually inject that plasmid into a plant. And so we have the main bacterial genome, but then we have this reduced circle of genetic material, the plasmid, which is available to be ejected out of the bacterium into either another bacterium or into a host plant. Well, one of the things that we have in our genetic toolkit, which we also got from bacteria, is there are enzymes. So remember that DNA is this double-stranded molecule. Adenine, thymine, cytosine, guanine, adenine, guanine, cytosine, cytosine, thymine, adenine, adenine. If this is the sequence of nitrogenous bases on one strand, what's going to be the sequence on the other strand? Adenine always binds to thymine, thymine always binds to adenine, cytosine always binds to guanine, C, T, C, G, G, A, T, T. So there are these enzymes called restriction enzymes. And they basically cut DNA at a specific site. And so maybe you have a restriction enzyme that cuts DNA at a CGAGCC site. And so what the enzyme does is it recognizes this sequence of nitrogenous bases in DNA. And what it will do when it recognizes that sequence of bases is it will come into that sequence of bases and it will break the bonds here, and it will cut DNA here and here, so that you end up with a DNA molecule that looks like this, with 3 nitrogenous bases hanging off here, and you end up with another DNA molecule fragment that looks like this. And so these things have free ends. And those ends allow you to add nucleotides to that. So this guanine wants to hook up with a cytosine nucleotide. This cytosine wants to hook up with a guanine. This thymine wants to hook up with an adenine. So because this restriction enzyme leaves these free ends hanging out, if you know that there's a gene somewhere in some other organism that does something useful, you can go to that organism, you can use the same restriction enzyme. If this is where the gene is located in the genome, this is the gene of interest, you can find a restriction site above that and find a restriction site below that, cut the DNA here and cut the DNA here, and then you have a free end here and a free end here that you can then use to put in this space that you've created, because this free end here is going to be a cytosine guanine guanine, so it will complementary. Whoa, I did that wrong. No, because I wrote it wrong. I wrote it wrong. Yeah. It's this, GCC. So this cytosine guanine guanine will complementary base pair to this guanine cytosine cytosine. This cytosine guanine adenine will complementary base pair with this guanine cytosine thymine. And you take this gene of interest right here, and you plunk it into this space in the plasmid of this bacterium. So now you've taken a gene out of one organism, you've put it into the plasmid of this bacterium. Now what this bacterium is going to do is it's going to, this is showing what I just showed you. What this bacterium is going to do, once you have your gene of interest into the bacterial plasmid, is the plasmid is going to then be injected by the so you put the plasmid back into the bacterium, and then the plasmid from the bacterium gets injected into the plant, because the bacterium is a bacterium that essentially takes this plasmid and inserts itself into the genome of the plant, and this is how it causes diseases. When it inserts this plasmid into the genome of the plant, it's going to insert this gene that you want in the plant into the plant. You do this early on in the development of the plant, and then because of the process of mitosis, mitosis just makes copies of the DNA that is in the plant, and it makes copies that are identical to the original DNA. The process of mitosis then propagates this gene all throughout all of the cells of the plant as the plant develops. So we take a bacterium, we open up its plasmid, we insert the gene of interest, then the bacterium infects the plant, transfers the gene of interest to the genome of the plant, and then that plant grows up with all of that, all of its cells of its body having that introduced gene through the process of mitosis, just this process of somatic growth. And this is traditionally how we have made transgenic plants, these genetically engineered plants. It's a little time consuming because you have to do all of the work of getting the gene of interest into the plasmid, but the fact of the matter is a lot of people work on the genetics of bacteria, and so we actually know how to do this pretty easily, so it's much easier to do it now than it was 40 years ago. And these are simple techniques. We have students in biology here at William Jewell College where part of their research, their senior research that they do for their degree is actually moving genes from one organism to another, inserting them inside plasmids, getting those plasmids into, for example, nematode worms. Rose Reynolds works on nematode worms, and you can actually, if you can get a gene into the genome of E. coli, so these worms eat bacteria, so you grow them on a petri dish, you grow bacteria on that petri dish, and then you put the worms on the petri dish, and the worms eat the bacteria. Strangely enough, the way their system works, the way their digestive system works, is that if you can get the gene into the bacteria, when the worm eats the bacteria, the bacterial genome actually will get incorporated into the genome of the worm. So it's really easy to get genes into nematode worms and transform them in different ways to study the effect of a particular gene on the development of the worm or the lifespan of the worm or whatever. She studies lifespan, actually. There's also a thing called a gene gun. It's basically a little air-operated gun, and you have your DNA fragments of your desired gene, and you actually use high-pressure air to essentially shoot them out of a little gun. They go through the cell membranes of the cells, and they get into the cell by basically just, it's called a gene gun actually, by essentially shooting cells with a gene gun to get the desired gene inside the cell. There are lots of ways of doing this. This is a relatively efficient way with plants because we've hijacked the behavior and the physiology and the genetics of a bacterium that already infects plants to then allow us to infect the plants with whatever genes that we want to infect the plant with. And then once we've done that, that gene is in the genome of the plant, and then that plant has whatever that trait is. This is not terribly hard to do in the 21st century. It was harder to do in the 1980s, but it's fairly standard nowadays in biology. Questions about this form of genetic engineering? We use enzymes to do all of this. So enzymes are cool because they do cool stuff like cutting open DNA. There are new things, though, that are on the market. Have you ever heard about CRISPR? Nobody? You've heard of CRISPR?
Mhm.
What have you heard of CRISPR?
The name of it?
Okay. All right.
I don't know exactly what it does, but.
Let's take a minute. This is probably the last thing we'll do today, and then next on Friday we'll talk about CRISPR in detail in terms of how it works. CRISPR has made news a couple of times in the last couple of years. It made news because a research team in China produced two human twins, one of which was CRISPR to get rid of a genetic defect, and one of which was left intact. And then more recently, this was back in July, a woman got CRISPR'd to get rid of sickle cell anemia, if I recall correctly. Yeah. And so sickle cell anemia is a human blood disorder. It predominates in places where there's a high incidence of malaria. So as a result, a lot of African Americans are plagued with sickle cell anemia. It's a 1 nucleotide mutation in the hemoglobin gene that affects the structure of hemoglobin. And so the idea is if you can go in using CRISPR and fix that 1 nucleotide, fix that hemoglobin gene such that that mutation is no longer there, hemoglobin will form correctly and you'll get rid of sickle cell anemia. And this woman volunteered to basically have herself CRISPR'd.
Now an NPR exclusive, the first interview with someone who has volunteered for one of the most highly anticipated medical experiments in decades. The first attempt to use the gene editing technique CRISPR to treat a genetic disorder in the United States. NPR health correspondent Rob Stein has our report.
I went to this research institute in Nashville to meet Victoria Gray.
Victoria's in room 7.
Hello, I'm Rob Stein from NPR.
Victoria Gray. Nice to meet y'all.
Victoria's just turning 34. She lives in Forest, Mississippi with her husband and 4 children.
My twins, Jaden and Jadasia, they are 11. Jamarius is 12, and my stepdaughter Asia is 13.
Wow, you got your hands full.
I do. This is the right time to get healed.
Victoria's been struggling with sickle cell disease all her life.
It's horrible. When you can't walk or, lift up a spoon and feed yourself, it gets real hard.
Sickle cell affects millions of people around the world, including about 100,000 in the United States. Most, like Victoria, are African American.
Victoria, this one is not gonna stop today.
A genetic defect deforms the red blood cells, making them sickle-shaped, hard, and sticky, and they don't carry oxygen like they're supposed to, causing terrible bouts of pain.
Sometimes it feels like lightning strikes in my chest and real sharp pains all over, and it's the deep pain. I can't touch it and make it better or do anything to make it better. Sometimes I would be just balled up and crying, not able to do anything for myself.
That sounds just awful.
Yes, it is.
The sickle cells have already damaged Victoria's heart, made her weak, prone to infections, strokes, and more. Many people with sickle cell can't work or finish school. Many don't make it past their 40s.
it's scary, and it affected my oldest son, you know, because he's older, so he understands. So he started acting out in school, and like his teacher told me, I believe Jamarius is acting out because he really believes you're gonna die.
So sorry. Some patients can get bone marrow transplants, but for most, there's no cure.
It was just my religion that kind of kept me going.
But then, Victoria heard about something new. Scientists were planning to use the gene editing technique called CRISPR to try to treat sickle cell for the first time. She jumped at the chance to volunteer.
Hey Victoria, how are you doing today?
I'm doing okay, Dr. Franklin.
That's great.
Dr. Hyder Frangoul is running the study at the Nashville Hospital.
How was your last night?
Last night, it went pretty well.
CRISPR lets scientists make very precise changes in DNA. Doctors have started using CRISPR to try to treat cancer, mostly in China. Frangoul's sickle cell study is part of a new wave of studies trying to use CRISPR to treat other diseases around the world.
CRISPR technology has a lot of potential use in the future, not only in blood disorders, in other genetic disorders to help many other patients.
Here's how CRISPR might help sickle cell patients like Victoria. Healthy red blood cells use a protein called hemoglobin to carry oxygen. Sickle cell is caused by defective hemoglobin, but babies, they're born with healthy fetal hemoglobin.
Once a baby is born, a switch will flip on. It's a gene that tells the red blood cell, the bone marrow cells that produce red cells, to stop making fetal hemoglobin.
So Frangoul will be using cells that scientists have edited using CRISPR to flip that fetal hemoglobin switch, to turn the production of healthy fetal hemoglobin back on.
And this opens the door for many patients to potentially be treated and have their disease modified to become mild or cause them no long-term horrible side effects of sickle cell disease.
Other doctors and scientists are also excited about studies like this. Lori Zoloff is the bioethicist at the University of Chicago.
This is an exciting moment in medicine. Everyone agrees with that. CRISPR promises the capacity to alter the human genome and to begin to directly address genetic diseases. But no one knows.
She worries the new CRISPR studies may not have gotten enough scrutiny by objective experts.
This is a brand new technology. It seems to work really well in animals and really well in cultured dishes. It's completely unknown how it works in actual human beings. So there's a lot of unknowns. It might make you sicker.
Dr. Frangoul acknowledges there are always risks with experimental treatments, but he says the research will go very slowly and carefully, with a lot of oversight by the Food and Drug Administration and others.
We are very cautious about how we do this trial in a very systematic way to monitor the patients carefully for any complications related to the therapy.
For her part, Victoria says she understands there may be risks. The treatment's not easy. It involves a grueling round of chemotherapy, weeks in the hospital, months of tests, and the study's just the first step that may only benefit other patients years from now. But she can't help but hope.
Being able to wake up and not be in pain, and to just be tired because I've done something and not just tired for no reason, and just to be outside and jump on the trampoline with my kids and go to graduations and weddings and see them grow up. That means the world to me.
It'll probably be months before the first clues emerge about whether CRISPR-edited cells are safe and might be working for sickle cell disease. And it may be years before anyone really knows how well they might help patients like Victoria Gray.
This gives me hope if it gives me nothing else.
Rob Stein, NPR News, Nashville.
So on Friday, I'll introduce you to how this actually works. I'll take you through what it looks like at a genetic level, how the CRISPR system interacts with DNA, and how you can use that to either, in this case, turn genes on or turn genes off or insert genes into places in the genome. And that's what we'll pick up on Friday. Get reading the GMO report and start talking to your team members, group members, about your plans for that.